Good morning or afternoon to you, and Happy New Year. Thank you for joining today's webinar. I'm Jan Reichert, Executive Director of the Antibody Society, and will be your moderator. This webinar is the first in a series of webinars designed to inform and educate our members and the broader scientific community about antibody discovery and development. Today's webinar is Antibody Discovery in the Cloud, using next-generation sequencing to expand the universe of selectable antibodies. Our speakers are Dr. Erasmus and Dr. Andrew Bradbury from Specifica. This webinar is being recorded. We will take questions at the end of their presentations, so please add your questions to the Q&A box in the viewer. Without further ado, I'll turn the show over to Dr. Bradbury.
Hi, welcome to this. Thank you very much for joining us. It's my pleasure to really be the inaugural speaker on this webinar series organized by the Antibody Society. And one of the reasons we tried to set this up was to just have a bit more communication, bit more interaction. It's difficult in these COVID times to really sit at home and I guess this is a big Zoom meeting, but anyway, we've got some exciting new results we wanted to share, and some of the other speakers will be doing the same. We're going to be trying to do this once a month or so. So what we're going to be talking about today is a little bit on the background of our Generation 3 library, but the main focus of the talk is how we are using NGS, next-gen sequencing, to expand the universe of selectable antibody. And what we'll be showing you is that by putting next-gen sequencing into your discovery platform, you really increase the number of antibodies significantly over just random picking. Even if your random picking capabilities can go up to 10 or 10,000 or even more. So talking about the Generation 3 library, this is a library design that we came up with a couple of years ago, and basically we're continually improving it, but basically the idea is that we start off with the best therapeutic scaffolds, and what I mean by the best therapeutic scaffolds are those that don't show any liability, that are well expressed, that behave well, have been in patients, and we think this is important. Then the next step is to isolate CDRs, and these are complete CDRs, either sequencing them and identifying them by next-gen sequencing, or alternatively by PCR, and I'll explain the difference between those two things in a second. We then eliminate all those CDRs from the NGS data set of CDRs that contain any sequence liabilities, and these contain ones that people know about, deamidation, isomerization, and so on, as well as some that we've discovered in-house in our experiments. Finally, those liability-free CDRs are embedded within the therapeutic scaffolds that we've chosen, and that gives us essentially a library within a therapeutic scaffold that has many different CDRs within them. So what this means is that we have combinatorial diversity, but not within CDRs. What scaffolds to use? So we actually have about 9 scaffolds that we've used to date. These are 5 of the original ones that we've used, and we still use these for many of the libraries. So the idea is here to have diversity in the V region. So you've got VH1, 2, VH3, VH4, and then you have the same V kappa 1, V kappa 2, V kappa 3, V kappa 4, and V lambda as well, which is combined with VH1. As you can see, most of these are a couple of humanized ones, and most are approved, and one in phase 3 and one in phase 2. One in phase 3 was removed because it didn't work as an antibody, but it still performed well in terms of developability. Now, the diversity strategies I've mentioned, all CDRs are from real antibodies. And the reason we think this is significant and important is because CDRs from real antibodies have come from B cells, and B cells will display antibodies on the surface. If the B cell is unable to display an antibody on the surface because it's poorly expressed, because it aggregates for any reason, then essentially the B cell dies. So the fact that an antibody is on the surface is an indication of on the whole of a relatively well-behaved. Therefore, the CDRs within those antibodies, which are contributing to the folding of the antibody, are all. So we use what we call replicated natural for all but HCDR3. These are extracted from our proprietary data sets. We eliminate errors and sequence-based liabilities, and then DNA is synthesized in arrays. And for HCDR3, we directly amplify from at least 1 billion B cells from at least 10 donors. And these will contain some liabilities because we're not screening these. And the reason is because the diversity, you just can't produce 100 million CDRs on a chip at the moment. And as I've mentioned, the recombinatorial diversity is between, but not within CDRs. One of the things we do throughout is next-gen sequencing to ensure the quality of our libraries, and this shows the progression of quality. So that Gen 1 library that's indicated there, that's a library I made about 20 years ago with Daniele Sblatero when I was working back in Italy. We thought it had a diversity just under 10 to the 8, so just under 100 million. When we did next-gen sequencing on it, turned out the HCDR3 diversity was actually only 3 million, so 30-fold less. The Gen 2A library indicated there is made from a single donor, and you can see the diversity is still increasing. Gen 2B made from 15 donors. You can see the diversity now is going up. We're now talking about 70 million or so. And the latest Gen 3 libraries that we've been making, we routinely get above 100 million, and as you can see in these two cases, a couple of 100 million different CDRs, HCDR3s. So all very well that we've designed it well, but how does it work? So our platform at the moment is to create the library in phage display. We do a couple of rounds in phage. We then transfer to Yeast in a single step cloning using homologous combination. And this allows us to fine tune affinity, specificity, and expression levels. And in particular, if you want antibodies, for example, that bind one target but not another, or you want an human and cyno, stuff like that. This is where Yeast display really becomes valuable. And then we do random clone pick, very much what everybody else has done. And you generally get more antibodies the more clones you pick. It's as simple as that. However, I've always felt that the true depth of diversity random clone picking isn't really being matched. And the only way you can do that is by doing next-gen sequencing to look at the full extent of a selection output. And then we use to organize the sequences, the antibody sequences that we get into clonotypes or clusters. And we then use clone picks, the randomly picked clones, as well as the next-gen sequencing and machine learning to identify a set of antibodies that we either have in hand if they've been picked, or if not, need to using gene synthesis. So this just shows how diverse the clusters that we're pulling out are. So this is a selection against GM-CSF. You can see looking at about 20,000 reads for a selection carried out at either 1 or 10 nanomolar. We're getting 1,000 approximate clusters for the 1 nanomolar selection and 3,500 for the 10 nanomolar selection. Question is how much do clusters differ one from another? So on the right-hand side, we have the differences in HCDR3 as measured by Levenshtein distance. What you can see is that the average difference between two clusters is 10 to 11 amino acids. So that's a big deal. We're not talking about amino acids that just differ by we're not talking about CDRs that differ by 1 or 2 amino acids. We're talking about CDRs that are really very different from one another. And if you take all the CDRs together, you concatenate them and you compare them, you can see we're getting differences of between 20 to 40 between antibodies in different clusters. Now, if you remember, one of the things we did in our design was to eliminate sequence liabilities. And so if that antibody generation 1 library, you can see that about 3/4 of the antibodies contain between 2 and 4 liabilities. If, on the other hand, you look at the generation 3, 3/4 of them do not contain any liabilities whatsoever at the sequence level. Of the remaining, most are found in HCDR3, and of those, they represent usually a single liability, and that's due to the fact that, as I mentioned earlier, we've amplified CDRs directly from patient B cells. One thing I should have mentioned, I forgot to mention, was that we only use, we synthesize libraries exclusively for individual clients. And so each library is exclusive to that client. We only used the B cells, the donors, once. We never use them again once we've made libraries. So what do the affinities look like? I'm not sure if you can see these or not, but these are some SPR plots using the Cartera LSA, looking at some of the antibodies that we've got here. And if you can read, you can see, for example, they're down here, I think, picomolar antibodies. There's a lot of subnanomolar antibodies, and the rest are basically between 1 and 10 nanomolar. And if we plot a bunch of different selections that we've carried out on an isoaffinity plot, then what you can see is that 20% of the antibodies we're pulling out, doing relatively standard selections in our hands. So 20% are less than 1 nanomolar, and 40% are between 1 and 10 nanomolar. So these libraries are performing really rather well, and in fact, I have to admit, much better than we thought. So when the pandemic came along, it was really to get involved and try and do some selections against COVID. And so what we did was we made a library from a patient who had an extremely high titer using immune kappa or immune lambda. And then we also compared to Gen 3. And we did selections against those 3 libraries. And here what you see is a yeast display plot. So in the x-axis, you have the amount of display. So each dot represents a yeast with antibody on its surface. The further to the right, the more antibody on the surface. And on the y-axis, you have the amount of S protein binding. And so the further up the plot is a yeast particle is, the more S protein it's binding, the more spike protein it's binding. And if you look here, in the middle at the 1 nanomolar set of plots, you can see that the generation 3 library is pretty similar to the immune kappa library, slightly less good, and much better than the immune lambda library. So cutting a long story short, we pulled out a bunch of antibodies. We tested their affinities, as shown as affinity plot here on the right, and also on the left. In orange, you have binding against the trimer. In blue, you have binding against the receptor binding domain. And what you can see is that the majority of these antibodies are subnanomolar. You can see the best we've got is a 15 picomolar antibody here against the receptor binding domain, and also another one which is binding the trimer at the same level. Interestingly, there's not a great correlation between the affinity for the receptor binding domain and the affinity for the trimer. In the isoaffinity plot on the right, we've plotted all the antibodies we've selected in blue, and then in orange are a few antibodies that Dennis Burton's team in the Scripps pulled out from a convalescent patient. And what you can see is that the affinities of these antibodies are actually, on the whole, in the same sort of ballpark. If anything, the Specifica antibodies might even be a bit better than the antibodies that have come from patients. We tested these for pseudoviral and live virus neutralization. I've got the results for pseudoviral neutralization. The live viral neutralization results are actually extremely similar. And so what you see here in red is, again, those 2 antibodies. These are the best 2 antibodies coming from the Scripps lab, generated by Dennis Burton. And then in blue are the various antibodies we pulled out, recognizing the spike protein. And as you can see, the pseudoviral neutralization titers are extremely good. Basically, we have a couple of antibodies here, less than 2 nanograms per ml. And interestingly, we found that the IC50 does not correlate with the affinity of the antibody. And you get a sense of that from looking at the table on the right-hand side. As I said, the IC50s of the live virus are very similar and in some cases perhaps even better than the IC50s we've got from the pseudoviral neutralization. So how do these antibodies compare to published antibodies? And so here are various plots from a number of different papers, as indicated here. And these are pseudoviral neutralizations. And what you can see, our best antibody is indicated in blue here. And you can see that in most cases, our best antibodies are actually to the left of the best antibodies coming out of some of these patients, with the exception of these 2 here. This is a bit of a surprise because the dogma with naive libraries is that you really need to affinity mature after you've selected. And what we're finding here is that the affinities coming straight out of naive libraries are pretty good. So now I'd like to briefly go through developability. So what we did was we took the single chains, we converted them to full-length IgGs. We then measured biophysical liabilities by various developability assays, as indicated here. We compared them all to the parental antibodies from which the clones were derived. This antibody at the bottom, sirukumab, which has very poor developability properties. You can see here size exclusion chromatography from clones from the first library, and on the left-hand side, you see the parental and the clone derived from it in the 4 plots. There's 4 different clones. You can see that these are all very, very highly overlapping. And in fact, the dark and the light represent untreated and 10 times. And again, you see very little difference between the frozen and thawed clone properties and the frozen and thawed properties of the parental antibody. So if you summarize all the developability results that we got, so sirukumab, as I said, is very bad, has polyspecificity, bad SGAC signs properties, and so on and so forth. The light blue, which represents antibodies within the therapeutic range, describes ipilimumab. As you can see in here, everything is light blue. But when you look at clones coming out from the library, you can see they're either as good as ipilimumab, which is a therapeutic antibody, it's gone into patients, or even slightly better in some cases. So summarizing all the antibodies that we tested this way, we find that over 80% of the antibodies have no biophysical liabilities. That is, they have no liabilities we can actually measure. Basically 3 antibodies or 16% have a single liability. We haven't found any yet with more than 1 liability. And so just to summarize this part of the talk, so these libraries based on well-behaved clinical antibodies, they're substantially liability-free biophysically and also sequence-wise. We have very high functional diversity, and we're providing these libraries, we provide these libraries exclusively. And then the properties are over 80% have no biophysical liabilities, over 60% have affinities less than 10 nanomolar. And in general, I would say we get between 500 and 5,000 different clusters per target. And with that, I'll stop, turn my camera off. And if you have any questions on this part of the talk, then please save them to the end. I'll now hand over to Frank Erasmus.
Okay, so coming downstream, I'm going to talk about how we're selecting and how we employ next generation sequencing to identify candidates kind of in a streamlined fashion in some of the pipeline that we incorporate and using this in our cloud-based discovery platform as well. So first, I just want to get into some of the limitations of standard selection campaigns, and particularly the problem of redundancy and sampling. So just some basic definitions up front. So a unique clone are sequences defined by greater than or equal to 1 amino acid in the CDR space. The clonotype is, in our case, that we define mostly throughout is a Levenshtein distance of less than or equal to 2. In the HCDR3 and LCDR3, we typically use PacBio, but this can be different. Clusters are basically our proprietary unsupervised machine learning approach where we take in consideration physiochemical properties of the amino acid space and any combination thereof. And this kind of condenses down H3L3 information in combination more significantly. So all in all, the number of clones is typically far greater than the number of clonotypes, which is greater than the number of clusters. So in an ideal selection, in an ideal scenario, you would pick 100 clones and you would obtain 100 unique antibodies. If this is a percent abundance plot, basically of their representation of the population, you can see it's all uniform. But this isn't the way that true selections work. 100 clones picked typically does not equal 100 unique antibodies because of the power law distribution of the clones. So you're picking a lot more from these more abundant clones in the population that can take all the way up to 80 to 90%, sometimes 50%, sometimes 20% in a given selection campaign. So it's really dependent upon the conditions of the selection, how hard you push it. But what this does is it biases these clones in the higher percentage range from a Sanger population. And while low throughput screening and next generation sequencing are going to perform very well when you look at this percentage range, if you get to the lower percentages, it becomes harder if you have, dependent upon the throughput, to discriminate against these certain populations. And in particular, when you start to get into less than 1%, especially if you're only in a single Sanger plate limit, you're going to be very limited in the total number of populations. And so if you're trying to discriminate a sequence from 0.2% or 0.01% that have been identified, again, these may be randomly showing up at given times. It's difficult to do this in Sanger, but in NGS, it becomes fairly easy to discriminate, because this is about 20 times more abundant in NGS, even though the frequency seems fairly low. Another problem that you face is sampling similar functional space. So as you know, high percent identity among the H3 and HCDR, or heavy chain and light chain sequences, exists across the CDRs. And based off the population, how you're conducting the selection campaign, there's certain features that are always going to come up, and it really is based off the population, the underlying population, the target that you're selecting against, how big the target is, and how many epitopes are available. But in any given case, oftentimes the higher percent identity across the space are going to share a lot of functional features. So this is an actual interference selection that was conducted, and you find a lot within the cluster space. And so if you move down here, each of these are plotted. The size of the circle is actually the number of sequences in a given cluster. You find a lot of patterns across here. And so when you're picking across here, you're actually choosing a lot of clones that may have similar paratope and corresponding epitope, and then perhaps functional similarities downstream. And so the third problem that you deal with is handling the data and processing. Many people are kind of, don't have the capabilities to work with NGS, or, it kind of can be cumbersome. You need expertise in order to handle this large data set. And so, just the mere fact of large processing and storage requirements becomes a hurdle. So to overcome these limitations of undersampling, high percent identity, or big data, we employ next generation sequencing. Obviously, this will fix an undersampling issue. You get far more bang for the buck. So you can get 2 million sequences from a PacBio, or up to 10 million sequences from Illumina, which is giving you a lot more sampled information. The percent identity is where we kind of employ our machine learning approach to maximize our selection and more efficient sampling across the CDR space. And then big data, we've incorporated with our collaborators, OpenEye Scientific, which is more focused in small molecules, to build a cloud-based platform that allows users to do this kind of work very streamlined and efficiently. So when we think about enhanced sampling, the two platforms that we predominantly use are Illumina and PacBio. For selections, we've been focused more on PacBio just because it gives us the paired-end reads, VH and VL, whereas Illumina is only restricted to a single chain typically. We find that PacBio for the selection outputs and the anticipated diversity is fairly sufficient in giving us the underlying sampling needed from a given selection campaign. We also take a targeted subregion approach, and there's many different strategies. Again, in this particular endeavor, we're exploring many different avenues, but this is one case example where we looked into the germline family assignment. Obviously, this is kind of standard of practice to assign the germlines to the family of V genes within databases. We also consider CDR1 and 2 separately in their corresponding VH and VL, but still use this information to build our cluster space. But then most important, we weight most heavily on CDR3s of the VH and VL. In particular, we focus on clusters and subclusters, where a cluster is basically a broader definition to try to capture paratope similarity. But then more refined clusters give us kind of distinction, maybe more biophysical distinction for affinities on rate or off rate. So any tag in this particular case that we'd have is a heavy chain scaffold, that's grouped with a cluster group in the HCDR3, and then a subcluster group to kind of have a distinguished pattern within this space. But the goal here ultimately is to maximize H3 and H3 plus L3 diversity in order to find certain patterns of interest within the population of interest. And when we employ our unsupervised machine learning to explore the CDR sequence space, what we find is that typically within the cluster space, so in these individual cluster groups, the median difference is around 10 amino acids that kind of captures this broad. And this is across merged CDR 1 through 3 of the heavy and light chain. But typically across each of these individual clusters, as Andrew kind of showed, this varies from one selection to the next selection. I believe this one is from the interferon selection that we're typically obtaining up to about a median of 40. This can drop down to about 30. Again, it's population dependent and varies based off the underlying diversity of that population and how broad and distinct the sequences are. Third solution is to build our cloud-based discovery platform. So not only do we have our, Specifica is basically experts in the field for building drug-like antibodies, and in particular through our exclusive libraries, where we can obtain 500 to 5,000 antibody clonotypes per selection, 20 select antibodies that are subnanomolar and greater than 80% of select antibodies having no biophysical liabilities. But we've also teamed up with OpenEye Scientific that specializes in really state-of-the-art software packages. They're more applied to small molecules at the current moment, but by teaming up, we're grouping to build a Specifica platform, and we have built it in order to use antibody discovery in the cloud. And this is built for a broad range of audiences. So it's for the less skilled user and the highly skilled user. So a bench scientist can use this, select the top 100 clones of interest that they want in their pipeline. This can move forward. It generates a gene synthesis list that can be provided directly to the gene synthesis provider of interest. Data scientists can also implement their own code. And so if you have a workflow that's very interesting and you want to write your own process, that's fully configurable. You have the option to do this too. So it's built for both worlds. So the platform itself that OpenEye had built is called Orion. And so the way that we are organizing our bioinformatics are into cubes and flows. And so a cube is basically individually configurable. It can perform its own distinct task, and you can write your own cube for a given task of interest. A flow is a complete network of cubes, and each run on their own CPU and GPU. And you can connect this basically in any order that you want according to your process flow. You can build new workflows, and it also connects to an interactive web interface that allows you to sub-select fairly easy downstream. So our antibody discovery package, when we package this into a complete workflows of cubes and flows, what we can have is our fast cube quality filter, and this is actually our pipeline of interest, is we can filter low quality sequences through fast cube quality. We have demultiplexing capability, so if you have different conditions, selection rounds, this is fully incorporated. So all you have to supply is basically an Excel worksheet, and all the demultiplexing is taken care of you based off however many conditions you want to test out. We have our own internal IG matcher annotation tool, which basically allows us to annotate both synthetic, semi-synthetic, and natural libraries across a variety of species and annotation schemes. So you can identify the CDRs of interest to you. And then this produces an annotated record. We move this into downstreams where we can eliminate bad sequences. These are sequences that you may have seen before, that you selected against, that you just want to avoid overall because you've already characterized it. Or it can eliminate those that have certain properties that you just don't want to have in your final output. One of the others is to capture scaffold diversity. So basically to broaden the germline capabilities, and then also our maximization, our CDR maximization algorithm, which enables us to explore the CDR space much more efficiently. After we identify, we can find round-to-round enrichment, log-fold enrichment from earlier rounds of selections to late rounds of selections. And then we can also incorporate low throughput. So if you have previously characterized hits, one of the things is, okay, you find a hit that has an epitope of interest to you based off binning studies. You can go back and cluster these sequences in the context of NGS because of that in-depth sampling, and allows you to do this more efficiently and effectively. We have the capability to eliminate liabilities downstream, and then all in all, we can take all this aforementioned information to make our predictions of the final output. So in a case study, what we have is basically using our cloud-based platform for SARS-CoV-2 antibody discovery. So just as a comparison, if we looked at the total H3L3 diversity captured by one Sanger plate, and we look at basically in red is the unique Sanger and NGS, and then in blue is basically the unique NGS overall. what we're plotting is a cumulative percentage of the clones as they're captured on the Sanger plate, and these are ranked according to their abundance. You can see that if you take the whole context of H3L3 only and look at the total diversity, you're only capturing about 1% in the grand context of the NGS universe. If you take another phenomena is that when you pick according to the power law, 80% of the picks are coming from the top 8 H3L3 pool. So that leaves about 15 to 20% from the remaining H3L3 pool that you're actually able to select from if you consider multiple plates downstream. If we consider sort limits, again, every organization has their own limitations on how much throughput that they can conduct. And the nice thing about NGS is you can output all of this into a single tube with a demultiplex inline barcode, and that can be submitted. But nonetheless, if you look at this, for this selection campaign in SARS-CoV-2, a single plate was producing about 20 H3L3 uniques identified. At 10 plates, we were capturing around 76. This is a simulated portion based off the output and sampled population that were identified. And then if we're going to the yeast sort limit of about 10,000, we were capturing up to 300. So obviously more sampled, you're going to capture more. It's just you have to do a lot more sequencing, and sequencing comes at a cost. So a single plate costing close to about $1,000 in the Sanger setting. Another thing, as I alluded to before, is clustering your low throughput output in the context of NGS. So, a Sanger profile, oftentimes if you only sample a few plates, you're just sampling too little to actually effectively cluster this. You don't have an idea of the broader context of the population and the underlying features that make up that population. So if you try to cluster, does this make up 3 clusters? Are there only 2 clusters here? It's not. It becomes fairly difficult to do. But if we consider, even in this case, we looked at the top ranked H3L3 in the context in the SARS-CoV-2. With the population of NGS, it becomes far easier to cluster these groups. So you can basically toss into your Sanger sequences, and in the backdrop of NGS, find out where these are fitting in the grand scheme of the entire population. And when we consider the cluster diversity captured, using this kind of similar plot of cumulative percentage and then the rank of the clusters, we can find that, again, you're subsampling a lot of the same population in both to a degree, but you're only capturing about 11% in this particular selection campaign of the H3L3 clusters that were sampled. That means 92% of the picks were actually coming from the top 6 H3L3. So there's a lot of redundancy here. You're picking from the same cluster groups that share a lot of percent identity. And that leaves overall about 8% from the remaining H3L3 cluster diversity to select from. And again, when you consider the limitations, if you consider a single plate, you're capturing about 9 clusters identified. Multi-plate, you're getting about 23, and the yeast sort limit at 52 clusters based off a simulated study. And then out here, you're starting to get more rare sampling, which obviously presents the problem of much lower confidence in it being an actual truly productive or effective hit. So if we simulated this and we wanted to understand, how many plates does it take, based off, what NGS would produce in a single run versus multi-plate sampling from a single plate. One plate is able to capture less than 1% of H3L3 diversity of a Levenstein distance of less than equal to 2, and only about 11% of the H3L3 cluster diversity. If we go out to 10 plates, we're still less than 5% and 27% of the H3L3 cluster diversity. At 300 plates, we're pretty satisfied, but again, considering a single plate costs close to about $1,000, is this efficient for the purposes at hand? You are approaching up to 80%, which is fairly good, but it comes at a higher cost. And obviously 500, you're approaching that 94% range in the H3L3 clusters. This is just a different way, table format of the previous plot. But at 100 plates, we approach about 61% of the cluster diversity and only about 20% of the H3L3 clone diversity. And so to linearly increase the number of clusters by traditional picking and Sanger sequencing, the number of picked clones must increase logarithmically. So it just kind of makes sense. The more you pick, obviously you're going to find more. So what's a more efficient sampling method overall? And is that sampling method producing real clones downstream? And so as I spoke of before, we've come up with kind of an enhanced picking strategy across the cluster space. And so obviously with random picking, what you have is a lot of redundancy. This is the Levenstein distance of clones. and there's a lot of overlap of sequence redundancy within this space. Our approach, we basically can pick across the cluster space top representatives from each cluster group, and then move that forward into downstream screening. And again, the next question becomes, can an antibody engineer do this? It sounds kind of cumbersome to, cluster sequences, put this through NGS, do the barcoding. And again, the cloud-based platform enables this kind of in a thought-free manner. So all you have to do is take your output from NGS, which is a FASTQ file. You input your barcode file. You have the option to select top 100 antibodies of interest. You can drag and drop this into our user interface in the cloud, and then basically set go, and it will maximize diversity according to that population, the underlying population, and eliminate biophysical or sequence-based liabilities of interest to you. And then basically will produce, sorry, I go back one, produce a gene synthesis ready format of VH and VL that is directly ready to be sent to your gene synthesis provider. So there's also the option to do a hands-on interactive interface. So in the same sense, if you have your output and you want to, basically, look at these clones in more detail, here's a true selection campaign. We actually overlaid certain NGS populations in the context of NGS only, and these were previously characterized clones in purple. Across the x-axis was the actual cluster identification according to kind of the scheme we laid out before, and then this is the number of replicates that were identified in the NGS population. And so we can easily select here and choose the most abundance, and and green would be some clusters that we haven't explored before, or certain sequences we haven't explored before, and then and maybe if this was a characterized cluster that had a lot of favorable features in downstream characterization, we can then choose diversity here in additional sequences that may be useful. In addition, we can go across the cluster groups and choose diversity across there, and that enables us to kind of explore this more efficiently and effectively. I think the last phase of this, which is probably one of the more essential features, is this is all nice. This is based off next generation sequencing. You can explore all these sequences, but are these actually true hits? And what I mean by a hit is typically in our hands is something less than or equal to 200 nanomolar in the as based off surface plasma resonance. And so this is just the details of a campaign against target X. And so when we consider our total hits, again, being less than or equal to 200 nanomolars, we can see that when we took equal numbers of randomly colonized, random colony screening, and then recommended hits from our platform, we can see that the random colony screening still did fairly well, producing over about 100 hits. The NGS universe, not considering those that were found in the random colony screening, improved this to well over 250 hits. And in fact, when we look at the total number of clusters is where the true advantage comes in, is because of the redundancy, you're actually just screening a lot of the same clusters in this particular space. Whereas with the NGS and picking across representative groups, we're enabling ourselves to explore the cluster diversity more efficiently and effectively, and whereby we can obtain, in this particular case, 151 additional clusters identified with 2.4 more times the picking of about 1,600 colonies. So in addition, the hit rate from our particular case in this selection campaign was fairly effective. We got close to about,
you missed I think you missed the slide, Frank.
Oh, sorry. Nope. I went back to it.
Oh.
Yeah. So in addition, so if we look at the percent hits, using our strategy across this, it was fairly surprising that we got a fairly high hit rate. So from our random colony screen, we traced those back. Our hit rate was closer, just above 60% of the unique clones. But in our NGS universe, we're approaching 90 to 93% within that NGS universe of clones across the representational space. And so if we look at the clusters now, again, the hit rate of functional clusters is actually far lower because you're not able to explore that cluster space as efficiently. in that random colony screening as well. And so one thing also that came up that we were interested in is, okay, if we consider the percent abundance, and again, this may vary from selection to selection, but if we consider the percent representation in the NGS and the corresponding to affinities, there wasn't much of a correlation, at least from this selection campaign, that related the representation in NGS and the corresponding affinities, which kind of suggests in this case that exploring the diversity to lower frequency ranges, the ones that may be missed at that higher frequency subset are effectively missed. And in fact, what we found is a lot more high affinity populations at lower percent representation in the GS. And our platform actually performed fairly well, again, in this particular selection campaign. This may not always be the case. Sometimes you'll have a lot more winners that may be representative. It's all about exploring that diversity space more efficiently and effectively. If we look at that same kind of this was interesting to us because what we have is we're plotting the percent abundance in NGS, and we find in this particular case kind of fulfills this U-shaped phenomena whereby you have the lower frequency clones that made up most of the predominant better affinity hits. But if there is a threshold, so as soon as you start to get much lower, the affinities drop off substantially, even though this whole group all has fairly good affinities overall in the grand context, but you're actually broadening your scope, so to speak. So in conclusion, Specifica's Gen 3 platform, as Andrew spoke of, generates 500 to 5,000 different clonotypes per target with excellent biophysical properties. Greater than 80% of testing antibodies have no biophysical liabilities, and high affinities, 20% or less than 1 nanomolar, 40% of 1 to 10 nanomolar. But dependent upon the selection of the number of clones sequenced, random picking identifies about 1 to 10% of the unique clones in 3 to 50% of the clusters compared to NGS. This is done after multiple selections using NGS in the backdrop. The Specifica Antibody Discovery in the cloud platform expands the universe 10 to 100 times for unique H3L3 clones and 2 to 30 times for H3L3 clusters, whereby about 93% of the unique NGS identified of the clones were functional in our subsampling population so far. 76% of these identified clusters containing clusters contain binding clones as well. All in all, to linearly increase the number of clusters by traditional picking and Sanger sequencing, the number of pick clones must increase logarithmically. So not so for NGS and the Specifica platform. There's as shown before, the cloud-based platform has a thought-free approach, fully automated. You can use the default settings to identify the number of desired clones from 48, 96, or 192, or kind of an expert user, whereby you can all NGS features are configurable, and you can make the decision within your group. And then lastly, interestingly, in this particular selection campaign, percent abundance and fold enrichment did not appear to be correlated with higher affinity. I have to acknowledge my wonderful team at Specifica, including Andrew as well. We have a great team over here, and this Andre Teixeira, who was really essential in not only the informatics, but also building the Gen 3 antibody library from which we can gather some of this wonderful data. Laura Spector and Kat Perea, who are recently joined on the informatics team. Laura's really delving into our deep learning capabilities and supervised machine learning, and Kat has been essential on some of the low throughput Sanger tools and everything else. Fortunata Ferrara and Camila, without them, we would not have any of these this data that comes through as well from these selecting antibodies. They take rigorous care throughout the process, and Sara D'Angelo, who makes everything run from A to Z extremely effectively, and also has built some of these incredible libraries. From the OpenEye team, from Jesper, Jeff, and Fred, it's been really great to build this platform overall. And with that, I think I'll open it up for questions to Andrew and myself.
Jan, you're having problems.
Sorry. It's like Here I am. No, I'm gone. Here I am. Sorry about that, folks. All right, we are ready for questions. First three came in during Andrew's presentation, so I will direct them to Andrew. How do you remove the liabilities before the library preparation?
So I probably went through it quite quickly, and I apologize for that. Basically, what we've done is we've sequenced probably 40, 50 donors, incredible depth. And using that NGS data, we've been able to generate data sets containing all CDRs in human antibodies in the 6 positions. So this allows us to take those sequenced CDRs and then examine them for liabilities and eliminate those that have liabilities. So this is purely an informatic thing. And so I hope that explains. As I mentioned during the talk, we can't do that for HCDR3 because the diversity of the HCDR3s is so much greater.
Next question, how do affinities from the Gen 3 library compare to the clones from convalescent libraries?
So again, I'm sorry for rushing this. The antibodies that came from the scripts that had IC50s of about, the best one I think was 6 nanograms per ml, those were antibodies that came from a convalescent library. And we directly compared those same antibodies. Dennis Burton kindly gave us those antibodies. We compared them directly with our antibodies in the same assay. And so the affinities that we're seeing are better, and the IC50s are also better.
Next question, how long does it take between obtaining the patient's sample and having the antibody candidate?
So if you're doing a library from a convalescent patient where traditional sort of VHVL and amplification, then that will take us about probably 2 to 4 weeks to build the library, depending on how deep and big we want it. However, if you're talking about just taking donors and amplifying the CDR3s from the donors, we usually use 10 donors to build the libraries. That building a library takes us between 2 and 4 months, about 2 months to build the library, and then it takes us another 2 months to quality control it by next-gen sequencing, selections, and so on and so forth.
Next question came in during Frank's presentation. Do you have an explanation for why your naive Gen 3 library performs better than patient-matured antibodies? Could this be explained by elimination of the self-epitope repertoire?
That's a good question, Renee. My explanation for this is that the diversities of our libraries at the genetic level may be as diverse as other people's libraries, although because we use next-gen sequencing throughout the quality control, we know that our diversities really are as big as we think they are. I think our big advantage is that the functional diversity is actually greater. So I think our functional diversity approaches the genetic diversity, which means that most of the antibodies in the population are actually functional. I think that's what's going on. Also, and the reason we have such a high functional diversity is, as I said earlier, is CDRs are collected within the context of B cells that are displaying antibodies, and we also apply some internal quality controls to the CDRs themselves to make sure that we're not using, for example, CDRs that just occur once that may be sequencing error and therefore contain problems with amino acids in the wrong places.
Next question, which criteria do you apply to define the top 100 clones from the 500 to 5,000 clonotypes?
So let me give a big picture answer and I'll let Frank delve into the details. So the criteria, the way we wanted to do this was to, for an antibody engineer, does NGS, presented with a massive file, no idea what to do with it. As Frank showed, you just upload that file, you then put it through the system, and it comes up with 100 antibodies that you should synthesize. So what is it about those 100 antibodies? Well, what we try and do is we try and get the maximum cluster diversity. So we get representatives of antibodies in as many different clusters as possible. So this, we believe, we're in the process of actually doing experiments to prove this, but we haven't quite got there yet. So this will give you the maximum functional diversity in terms of epitopic binding. As Frank said, each of those things are if you're an expert bioinformatician, you can go in and modulate each of those individual components. Frank, do you want to say something else?
Yeah, absolutely. So in addition, so with the 100 is to kind of give you, so in addition to the 100, you can also pre-select, the number of representatives or the max number of representatives you'd want across that cluster space. So again, you may want to have representatives of up to 5, or you want to do it according to their distribution. So those options are available, or you can be more uniform. And we're approaching this from a uniform perspective, where we try to broaden our scope by selecting uniformly across those populations of interest.
Next.
Thanks for that question, Emmanuel.
Next question is, how do you address the artificial diversity introduced by the lower accuracy of PAC biosequencing?
So this is a great question. So we do several different steps. One, we set extremely high thresholds from PacBio, because you're absolutely right. And again, our underlying diversity is going to be from selection campaigns. You want to have a pretty good fold coverage of the underlying diversity that you anticipate. And so after that, we undergo 2 particular steps. One is basically an error clonotyping, which is standard across it, which is the informatic processing, where the more abundant clones based off kind of a string edit distance will absorb the less frequent clones. And this takes care of a lot of the small differences that you may absorb at the DNA level. And we do this at the amino acid level, which really, overcomes that across the diverse CDRs. And then again, the clustering itself is actually a way to overcome this as well, because within each representation, you're actually taking clones that have higher frequency, and those that are going to be more standardized across that in that given group. It's a great question.
Next question is, have you used the platform to select antibodies against glycolipid antigens?
Yes, we have, and it's not easy, but it does work. I think the more difficult component is directing selections to particular portions of a glycolipid, but we can definitely get binders against glycolipids.
Next question, why do Specifica libraries generate such high affinity binders? Is it just through high diversity, or does removing liabilities indirectly improve affinity?
So as I mentioned before, I think it's very high functional diversity with very carefully curated next-gen sequencing confirmation of genetic diversity as well.
When you try and maximize through clustering, what types of parameters make up your principal components? Are they primarily sequence features, or also binding, functional, or developability characteristics?
That's another great question. We explore a variety of them, and some I'm not privy to answer, because of proprietary aspects. But basically, we are exploring a variety of approaches that consider going beyond just the sequence-based amino acid so you can use it kind of a conversion space into physiochemical properties and consider certain features within there that can allow you to estimate kind of amino acid properties. But then also taking kind of NMER concatenation, or converting this into kind of a one-hot encoding, which works very well for, obviously supervised approaches. So we explore a whole variety of them. In this particular case, sometimes the most simplistic one is actually ones that perform fairly well, but obviously delving in deeper into these conversions, it's still something we're exploring.
Do the antibodies tested for developability come from pick colonies, so the most frequent, or from additional hits identified by next generation sequencing?
Another good question, Emmanuel. So it's basically a mixture. So when we were doing these analyses, we were keen to get antibodies from each of the individual scaffold libraries. And so some of them came from clones that we'd picked that we were able to show bound, and others came from next-gen sequencing. analyses, and so those were not they were identified by an NGS. So it's a bit of both. Is the answer.
Next question, how is the abundance defined in next generation sequencing? Is it just raw count, or is it based on some enrichment normalization? If it's normalized, do you think it will have more correlation with affinity?
Again, I think the correlation with affinity, we do normalization of enrichment as well as converting this into kind of percent abundance, which is going to vary across the populations using some of the read counts. And both in this case did not correlate very well. But I'm not to say, I mean, the way this was conducted, this particular selection campaign, does not necessarily mean it will relate to different selection campaigns whereby you'll have much better correlation. There were certain factors that may be included which may have not had that in this particular scenario. It varies very differently, at least how we conduct from selection to selection, the conditions and everything else. And I'm sure that in other cases, you may find far better correlation. Not in this case, though.
Next question, how do you cluster the sequences by CDRH3 or L3 plus H3, and what is the sequencing depth from PacBio?
So that's a great question. So again, we try to consider the entire H3, L3 as a predominantly weighted. So we want to maintain the paired information. We want to consider the germline information as the scaffold call, but we also want to consider CDR1 and 2 as well, but those are basically grouped. So in priority, it's basically HCDR3 followed by LCDR3, followed by the CDR1 and 2, followed by the germline scaffold call. And so all of that plays a critical role because obviously the scaffold is going to shift the underlying characteristics of the CDRs that may change slightly. So obviously we find that information very important. The sequencing depth of PacBio for Sequel 2, so they came up with new technology, and this to address earlier questions, is it's improved drastically. So Sequel 2 gives you about 3 million overall consensus reads after you, and then this kind of gets broken down, and based off how high you set it, you can obtain up to about 1 million sequence reads for VH and VL.
Can you discuss whether your libraries perform well for GPCRs or ion channels?
Hi, Vinod. So, sorry, wrong question. Sorry, Arjun. We haven't done selections on GPCRs and or ion channels. Very keen to do them. So if anybody's interested, we'd be happy to work with you on that.
Nice talk. Hopefully that is from all of the attendees. But the question is, did you find any VH or VL only chains, that is single domain antibodies? If yes, did they get tested?
So usually what happens is when we do the PCR, we're PCR amplifying full length chains, they go into yeast. However, we do find some VH and VL only chains, but we don't take them forwards. Perhaps we should, but right now all we do is look at the full length single chains of VH plus VL.
So this is someone picking up on the answer you gave about the liabilities. So thank you for your answer about the liabilities. But after the in silico selection of the CDRs without liabilities, how do you make sure they are not in the phage library? Are you synthesizing your full library from that selection? If not, how do you remove the liabilities from the phage library?
Sorry, I'm obviously not explaining myself terribly well. So we have a database. Let's think that we're looking at, I don't know, HCDR1, okay? So we have a database of HCDR1 sequences, which is divided into different VH families. And now that database of VH, of VH-CDR1 sequences, we examine that, and let's say there are, I'm just making up a number here. Let's say there are 10,000 sequences. And now we eliminate from that all the CDRs that contain isomerization, single cysteines, so on and so forth. So all the liabilities. And now that set of informatically identified 10,000 sequences now goes down to, let's say, 4,000 sequences, okay? And now, so what we've done is we've eliminated physically from our oligo pool the 6,000 sequences that have liabilities. Now what we do is we take those 4,000 sequences, and those are the ones that we synthesize. And we have CDRs for all the individual CDRs are synthesized except for HCDR3. And we take all those CDRs and we assemble them together to make full-length antibodies, including HCDR3s, which come from the natural patients, from the B cells. So if that doesn't answer your question, please ask another one just to make sure.
So we just have a couple of minutes. I think we can zip through the last couple of questions. The next one is for CDRH3 amplified from 10 to the 9th, I think that is, B cells. Are these pan B cells or specific B cell subgroups?
So right now, we use leukopacs to generate our B cells. The reason we do that is they essentially contain all the circulating B cells in an individual. And then we purify the B cells using antibodies against CD19. It's a kit that Miltenyi provides that we helped in the early development of. And they work really, really well. So yeah, so it's basically a pan B cell.
Yep. Okay. So that may mostly address the next question, which is, are your donors healthy? Which B cell population are you using for your library, naive, memory, or plasma?
So the donors, we choose healthy donors, but if somebody wants a library made of unhealthy donors, we'd be happy to make that. Well, actually, healthy. I mean, some of them smoke, some of them quite obese. So, you know, whether they're healthy or not is another matter. But they're ostensibly healthy. Which B cell donors are you using? Again, they're mainly naive and memory.
Okay, I think we can get one more question here. Can you rediscuss Sanger versus next generation sequencing in terms of sequence coverage?
And hopefully I'll address this right, but so Sanger typically, I mean, the way we think about Sanger is on a plate colony picking, and so 96 at a time, so close to 100 at a time, and then every single plate is equivalent to about it's about 850 up to $1,000 depending on how you look at it, and then NGS. Again, for MySeq, you can get up to 10 million, reads. It's about 24 million paired-in reads, and so that condenses down for single sequences into about 12 million or so, and then that condenses down when you filter for quality. PacBio again is around 2 million reads in total for Sequel 2, but again, this drops down as you filter out the low-quality scores and really retain the high subset as we do. Into about the upper hundreds of thousands to low millions.
Okay, I'm gonna push us a tiny little bit because there's just one more question. This completion thing, I gotta get it in here. Have you seen trends in the different scaffolds? Is there a scaffold that performs better?
That's a really good question. So we have seen trends, and it's all very, very antigen dependent, and we don't quite understand what's going on there. So, for example, some targets really like the VH1 scaffold. Other targets really like the VH3 scaffold. That said, that happens when you mix the libraries together. So if you mix all the libraries together, you select against a particular target, usually what we find is dominance of one particular scaffold. However, if you take the scaffold separately and you select against each scaffold separately and independently, then we tend to get binders from each of the scaffolds independently. So there is a bias, but that bias can be overcome if you use these scaffolds singly and independently.
All right, with that, thank you so much, both Frank and Andrew, for such a great, really interesting, informative presentation. I enjoyed it. I'm sure our attendees did also. To our attendees, thank you very much for joining the webinar today. An on-demand version will be available in a few days. I'll send a link to this on-demand version by email to everybody who registered. Please feel free to watch this or any of our other on-demand webinars when it's convenient. Thanks again, and have a great day.
And if there's any other questions that anybody has that we haven't addressed, then our emails are on this slide. Please get in touch with us, and we'll be very happy to get back to you.
Absolutely. Thank you.
Thank you.
Bye-bye.